I would like to, Belle, just stop there. Thank you, Belle, for all the work you did for organization. You always escape, so I wanted to capture you inside too.
All right, I'll be back around.
All right. So we are running a little bit short of time, and not to miss any minute of our next keynote speaker, I would like to go fast through a few of the challenges that we are cooking up for next year. So the challenges that we are doing, some of these might or might not happen. I think hopefully all of them will happen. There will be maybe other challenges as well. But as we calibrate our work towards the new challenges, sometimes there are hiccups, sometimes there are new opportunities. I will discuss basically 4 of these challenges: the NCI CPTAC Proteogenomic Challenge, the Malaria Dream Challenge, the NeuroLinks Dream Challenge, and the Multiple Myeloma Dream Challenge. So the NCPTAC Challenge, that is done in collaboration with the CPTAC, the Clinical Proteomics Tumor Analysis Consortium, with the help of people from CPTAC, like David Fenio, as well as Julio Saez Rodriguez from Aachen University, Paul Boutros from Ontario Institute of Cancer Research, Henry Rodriguez from the NCI, and a little bit of help from myself. So the idea is that cancer is, we are very familiar with analyzing cancer at the transcriptomics level, but there is a new push, especially from the CPTAC world, in trying to look at it from the proteomics level and to try to do it in an integrated way. The CPTAC folks have been analyzing the same tumors that were analyzed by the TCGA, looking at its genetics and genomics, transcriptomics profile. The CPTAC folks try to look at 3 of those cancers that TCGA analyzed: breast, colon, ovarian. And furthermore, they are doing 100 more cases of each of these cancers to have further data. So in total, it will be for these 3 cancers 200 samples, 100 of for each of the cancers, 200 samples, 100 of which are public, and 100 are going to be new. And so everybody is more or less familiar with the fact that we don't see a lot of correlation between the RNA and the protein corresponding to that RNA. So the average is about 0.39 in terms of the correlation. Some correlations are negative, some are not better than 0.39, but in average is that. So the question is, can we do better than that? And that's one of the questions. So the questions that we are thinking of, and this is an ongoing discussion, depending on how the data allows us to ask these questions, is going to be, can we say something about patient stratification, with the unification of the proteomics and genetics and genomics information? Probably we will not have statistical power to ask these questions, but we are considering it. Can we use genomics, genetics and transcriptomics to predict proteomics data, so to increase this 0.39 into a 0.8, maybe correlation level, and improve protein identification and quantification using genomics, transcriptomics, and proteomics data, including can we improve on missing data and imputation. So this is a challenge that hopefully is going to take place in the spring, starting in the spring of 2017. The Malaria Dream Challenge I mentioned a little bit earlier is a challenge that started at the lab of Mike Ferdick from one of our students turned now faculty in IBM South Africa, who is interested in malaria. And so the question there has to do with the fact that a drug called artemisinin is we are starting to see development of drug resistance to it. The causes of drug resistance are unknown, and there are no good markers to determine whether a parasite is going to be resistant. And so the question is, can we identify drug combinations that when administered along with artemisinin can combat better the disease. And so there will be two challenges. The data for this is pretty much all collected. Question one is, can we predict the IC50 from the artemisinin IC50 from transcription data, given that you learn some of the IC50 from known strains, but now you have a few strains for which you don't know the IC50 of artemisinin in those strains, but you know the genomic and genetic information. And the other one is, can we predict genes of importance in resistance to artemisinin? And part two is, can we find which drugs are going to be synergistic with artemisinin in killing the parasite? So stay tuned for this. Probably is going to be one of our challenges in 2017. There's another challenge that we are working in collaboration with the NeuroLinks community. And in this challenge, basically the idea is, can we identify progression in ALS patients? And this is pretty image intensive and probably very heavy in terms of data as well. There will be induced motor neuron differentiation grown, and the cells are going to be grown with different fluorescence tags. There will be microscopy that will look at individual cells as they grow, and a multi-channel image data will be generated. And the questions that we are asking are, can we develop algorithms to extract features from images that predict survival at the single cell level, and that distinguish cells of ALS patients from those of healthy people? So we want to do really from different, immune stem cells, inducing stem cells and then differentiating them into motor neurons, say something about the disease of the patients with ALS. Very good. And the last challenge is a multiple myeloma challenge that I will ask Justin, who's organizing it, to tell us about it.
In between you and lunch, so I want to quickly introduce the Multiple Myeloma Dream Challenge. The goal of this is to identify high-risk patients that are newly diagnosed with multiple myeloma. So this is the fourth challenge that is related to prediction of cancer outcomes. So previously we had the prostate cancer, the AML, and the breast cancer prognosis challenge. So if you're not familiar with multiple myeloma, this is a blood cancer related to B cells and caused by the malignant plasma cells in the bone marrow to proliferate. 25,000 newly diagnosed are diagnosed per year in the US, with 11,000 deaths. So there's obviously a clinical utility for a high-risk predictor. This includes the fact that early first relapse is still one of the strongest predictors of poor overall survival. And there is a clear need to identify aggressive disease to offer the best treatments and clinical trials for patients. So there are existing high-risk markers. Most of these are okay, I somehow can't see it here anymore. So these are high-risk markers that are based on beta-2 microglobulin, albumin, LDH, or cytogenetic features. High risk is often defined as the top 20% of patients with poor outcomes, which has a median OS overall survival of 2 years. Gustavo, I don't know what happened here. There we go. A number of groups have developed and looked beyond lab values in cytogenetics and created gene expression signatures of high-risk patients. Here's a number of them that have been published, and they all have different representations of what they consider high risk, leading to different proportions. We intend to use all of these as benchmarks, and we'll be evaluating these as part of this challenge. Part of the motivation is, in fact, that many of these gene expression signatures don't correlate very well. And so so their ability to identify a concordant set of high-risk patients is not very strong, begging the question, is there a better classifier that can be developed that is more robust and generalizable across multiple data sets? So here's the essence of this challenge, which is to predict high-risk patients both using two criteria, PFS and OS. And I think what's unique about this challenge versus work that's been done previously is really the size and the multiple assays that will be available. So we'll have available to solvers patient demographics, clinical history, laboratory data, cytogenetics, somatic mutation, and gene expression all together. So we've been going through the process now of aggregating these data sets and beginning our own quality control process as we do with all DREAM challenges. Here's a snapshot of some of the data sets that we have in hand. And this, from the genomics perspective, there's multiple modalities, including gene expression arrays, RNA-seq, and whole exome sequencing. And you can see some of the survival curves comparing across studies there. So to pull this off, we've had to engage multiple partners, and I'm very happy to report that these are the institutions that will be contributing data today, potentially others in the future. This includes the Multiple Myeloma Research Foundation, Dana-Farber Institute, the Moffitt Cancer Center, University of Arkansas Medical Center, and I would like to thank our challenge sponsor, Celgene Corporation. And similar to some of the other challenges that you've seen, we're going to take a Dockerized model approach where people can train their models and submit a Dockerized container, which we will then validate on data sets that participants don't have direct access to. So I'm happy to announce that pre-registration is now open. You can find it here on the DREAM Challenges website or at this URL here at synapse.org, and we expect to launch this in January 2017. Thank you.